Hello everyone. Sorry for the I took a lot of time. So today I'm gonna present about the shooting genes and proteins from a gun, ballistic biodelivery materials and methods. So can you guys hear me? And
Yes,
okay.
we can.
Okay. So when we talk about the vaccination, the most common and widely used methods for the vaccination is needle and syringe. Despite their common usage wide and common usage of this needle and syringe, there are several limitations associated with this method. The main problem is needle phobia because it's painful, and therefore, looking beyond this needle and syringe, researchers have done to uh, find new techniques by avoiding these needle and syringe to find new techniques without pains. So here, these are some of few most promising methods of vaccine delivery, which are painless. So therefore, researchers have done in this field to optimize the vaccine delivery uh, conditions and find new techniques to deliver vaccines with a painless manner. So some of them are gene guns, nano patches, laser adjuvants, microneedles, and edible vaccines. So in nano patches, they are used some dried coatings with macroprojectiles. When increasing the macroprojectiles, we can increase the amount of vaccine that can deliver into the body. Also using laser, we can also deliver vaccines with a painless manner into the body. Microneedles is also a common way to and also wide studied area of delivering vaccines into the body. And edible vaccines, in this method, the gene guns are directly involved in this process as well to make transgenic plants with the preferred gene that we want to make. So in this work, I'm going to talk about deeply about gene guns. When we talk about the guns that are used to deliver vaccines, they are called liquid jet injectors or jet injectors. So according to these images, left side figure shows some commercially available jet injectors, handheld jet injectors that are used to deliver vaccines. So this image also, right side image show the schematic diagram of the jet injectors, which shows the main parts consist of these liquid jet injectors and the schematic diagram of how it's going to work. So if we talk about how these liquid jet injectors are going to work, when the pressure applied to the liquid, here again, this is the liquid reservoir. Here we reserve the liquid vaccines that we want to deliver, and using this release button we can apply pressure onto that stored vaccines, and through this pressure we can eject out this jet through this nozzle in very high speed, like 100 meters per second, that can successfully deliver these vaccines inside the body, intramuscular and epidermal area. So researchers have done to identify the process and the factors affected to these jet injecting process. So in that, according to this left side image, it shows the stages of how this jet injection process is going to work. So according to the figure A of the left side image, it shows the jet injector is activated and ready to release a liquid jet with a high speed, like 100 meters per second. So with the applied pressure, the jet is coming out from the nostrils with a very high speed, and when it figure shows the first impact of this jet with the skin. So as soon as this jet impact with the skin, it makes a holes on the skin through erosion or other cell failure mechanisms. And through this hole, the liquid vaccines is going through the targeted area in a successful manner. So according to this research, they have found that 90% of these reserved vaccines can be successfully delivered into intramuscular or the targeted tissue that we want to deliver these vaccines. Also, in this research, they have
Did you say intramuscular? It shoots all the way through the skin into the muscle?
yeah, in jet injectors, they said epidermal and intramuscular level, they can deliver these vaccines. Yes, they said like that, intramuscular or epidermal.
Okay.
Okay. Then they have optimized the conditions. They have checked how the factors of the jet injector affect the volume of the vaccine that we can inject into the body. So through that, they have observed that when increasing the power of this nozzle from 1.5 watt to 37.1, what they have observed is when increase the power, they can increase the volume that deliver into the skin. When it comes to 37.1 limit, they conclude that they can deliver more than 90% of the reserved vaccines to the body successfully. So the power and the size of the nozzle also matters. So normal size of the these nozzle that they are using is like 76 to 360 micrometers. That is smaller than the standard needle. Outer diameter of the needle that is used, that outer diameter of the needle and syringe method, this needle diameter is ranging from 800 micrometers. So this is a very small than that. Also, these jet injectors, depending on the number of immunization shots per one reservoir, we can divide these jet injectors into two groups. They are multi-use nozzle jet injectors and disposable cartridge jet injectors. So in multi-use nozzle jet injectors, they used lots of repeated number of immunizations per one reserve vaccines and using the same nozzle. So earlier, this method, multi-use nozzle jet injectors method, have been widely used to treat diseases such as influenza, cholera, HIV, hepatitis B virus, like that. But suddenly, the use of these nozzle jet injectors have been stopped because they have found that by using the same nozzle, it can cause subject contamination diseases. So this is the major drawback of these multi-use multi-use nozzle jet injectors, and therefore World Health Organization restrict to use these jet injectors in human application, but still these are used in some veterinary applications. So, as I said earlier, the major drawback of these multi-use nozzle jet injector is the contamination of the nozzle with some liquid splashes. So, researchers have done to examine this process, how this nozzle is going to contaminated and it's caused to spread the diseases. So this image shows the snapshot of high-speed video of how liquid jet injecting process of water into human skin. So according to this time is equal to 0. This image shows, all of these images, the top black layer shows the outline of the nozzle. So it's the outline of the nozzle, while the black layer of the bottom, at the bottom of the each image, shows the outline of the skin. So in between this nozzle and skin, there is a few millimeter gap. So when at time equals to 160 microseconds, they observe the first impact of the jet on the skin. As soon as it impacts on the skin, they observe some liquid splashes coming off, bouncing back from the skin to the nozzle. So they have calculated that splash liquid volume at t equals 100 microseconds, that volume was equal to 100 nanoliters. So they conclude that through this process, the nozzle can be contaminated with the liquid splashes coming out from the skin and may causes diseases spreading. Therefore, this method is not currently using much. So the usage of this multi-use nozzle jet injector method is currently restricted until all the safety issues has been resolved. So moving on to the next part, the guns that we are using to deliver solid materials in a solid manner, so DNA, protein, other biomacromolecules in a solid state, that gun we commonly call gene guns. So this handheld gun called Helios gun is commercially available gene guns produced by Biorec, and that instrument itself is highly expensive. So this middle image shows the basic components of a gas gene gun. Inside of this gene gun, this is the process that's happening inside this. So it's mainly composed of the gas acceleration tube and also rupture disc. These rupture discs are also commercially available. They can withstand up to some pressure, but the pressure inside this gas storage exceed that level, this disk is going to rupture and allow this high pressure gas come out and to accelerate this micro carrier attached to the macro carrier through the stopping screen. Then these macro carrier attached to this micro carrier particle attached to this macro carrier leave the macro carrier with high speed and successfully transfect into the plant tissue. So this gene gun still use by now still using to directly transfect the preferred DNA and genetic material, also protein materials directly into animal tissues and plant tissues. So depending on the process of the operation and also the driving force inside the gene gun can be divided into main three types. They are powder gene gun, the first one. This is the most primitive design of the gene gun, firstly produced to transfect plant cells with preferred genes. So in that powder gene gun, in that instrument, they use ignition gunpowder as a driving force to give the accelerate these microparticles attached. Microparticles are the particles that we are going to attach DNA and our biomacromolecules onto these microparticles. So they're very small in size, and those microparticles attached to a macro carrier. So after ignition of gunpowder, it allows these macro projectiles to move through these gun barrel with a high speed. But when it comes to forward, this screen is stop this macro projectile, but the small microparticles leave from this macro projectile with a very, very high velocity, go through these holes and successfully transfect the cell culture that we want to transfect with genes of interest. So the main drawback of this method is the ignition of gunpowder can have a very high pressure and it can damage the target tissue culture. So damage is high, tissue and cell damage is high by using this ignition gunpowder. So Shang and co-workers, they have worked together to improve this primitive powder gene gun instrument by using a ground slide, by modifying it with including a ground slide, baffle plate, and by changing the gun barrel, also the base of this gene gun. So what happens here is when they also use ignition gunpowder as a driving force to accelerate the microparticles, but because of this using of this ground slide, they reduce the amount of pressure coming out from the screen and reduce the cell damage. because this bullet is going to stop by these ground slides, and also the microparticles coming out through this ground slide, they can come out through ground slide, and also by going through this baffle plate, they can successfully transfect the plant. So the next type of gene gun is high voltage electric gene guns. In that method, they are using high voltage to vaporize water droplets, and that by vaporizing those water droplets, that expanding gas act as a driving force to accelerate these microparticles into high speed, and also deliver genes as the same manner as I told you earlier. So the main drawback of this method is the high voltage is needed, like 18 kilovolt high voltage needed to vaporize these water molecules. So it is not very cost effective, and some other drawbacks are there. Therefore, this is not very commonly used, but some researchers still doing to modify these voltage electric gene guns using, they are trying to use limited voltage, and they are using different types of liquid instead of water to vaporize it and to get a maximum pressure, but still they are in research. The most common and widely used method is the gas gene gun. Here, the main components of this gas gene gun is air storage house, also a membrane. Here is the bullet. This is the left side image shows the before the bio delivery method. So before the process and after the gene transfection process, the figure right side figure shows the after the process. So before the process, membrane is still here and bullet is at this position. So when the pressure inside these air storage house exceed that the pressure that can withstand by the membrane, the membrane is going to rupture, and the bullet is going the pressure is coming down, and bullet is accelerated and impact with this baffle plate. So baffle plate hold this bullet but not holding microparticles. So microparticles with a very high speed can transfect the culture media that we want to transfect. So that biolistic delivery process can be divided into 3 stages: acceleration stage, separation stage, and deceleration stage. So acceleration stage happens inside the gene gun, as we discussed earlier. So the separation stage start from at the point where microparticles leave from the macro projectile. That's where the separation stage start and end when impacts on the skin. So after into the skin, the microparticles going through the
Yalini,
yep.
I have a question.
Yes.
Is there a microparticle size limit for the gun?
Sorry, what's the mic? You're asking what is the size?
Yeah, like is there a limitation on how large of a...
Yeah, that's I'm going to discuss in a few later. The
Okay.
size of the particle matters. Yes, I will discuss it. Okay.
All right, thank you.
Okay, then deceleration start when the particles impact on the skin, then they are going to inside the cells, and until it meet nucleus or targeting area, the process until that is called deceleration state. So as you asked, the size and the material that we are used to make microparticles or beads matters for this process. If we used large particles, they are in the deceleration process, they are used to take extracellular path. Extracellular path, in this path, what happens is as the particles are big, they cannot go in through cell membranes into each cell. So instead, they are weaken the cell boundaries and go through the cells. So on the other hand, the small particles that is size ranging from the like 0.6 to 6 micrometers, that is lower than the normal plant cell size, they can successfully go through the membranes of each cells and can successfully access to the nucleus and other organs inside the cells. If we talk about more of these two processes, extracellular process, large particles of low density materials are used. low density materials like stainless steel, polystyrene, and glass. When we talk about the glass, silicon materials, so they are very like low density materials. So the particle size of the material that they're using is ranging from 15 to 99 micrometers. So that's a big. Because of these large particles, the main drawback of that extracellular path is lots of cell damage and destroy can happen to the skin surface because of the size. And therefore, intracellular path is preferred, so particle-mediated DNA immunization. For this intracellular path, it would be better the particle as smaller as possible. So small particles of highly dense materials are used for the DNA delivery, DNA immunizations. The materials, most commonly used materials are gold particles and tungsten particles. So compared to the compare gold and tungsten, gold is preferred, but it's kind of expensive, but its biocompatibility, low toxicity, and it has no long-term toxicity or something bad happens. So gold is the preferred material for this immunization. Tungsten is cheap and also has very good advantage, but it has proven that it can cause toxicity in long term. After applying, long term toxicity can happen. And also, these tungsten particle, I read some research, they can induce, like, they can cause acidity inside the cells, so it can affect the processes. So, gold is a preferred material. So after the coating, in this process, if I summarize what they have done, they take DNA and coated them on the gold microparticles or other any preferred microparticle. Then after these coated DNA coated gold particles, we can successfully transfect inside the cells of plant and animals. So let's move on to the research that people have done to optimize these ballistic biodelivery process, as well as they have tried different materials as the macro carriers to optimize these processes to get the optimized immunization. So in this first research I'm going to talk about, they have used mesoporous silicon nanoparticles to deliver DNA and also chemicals into plant cells. So in this research, briefly what they have done is they have prepared 4 different types of mesoporous silicon nanoparticles. So in the first type, mesoporous silicon nanoparticles is like this. The structure, they have hollow, like tube inside the mesopores. To these mesopores, with the help of a linker, this is the linker molecule, they attach fluorescein dye. So they loaded a fluorescein dye into the mesoporous silicon nanoparticle. That is the type 1. Type 1 silica particle that they have made by loading fluorescein molecule inside the mesoporous silicon nanoparticle. Then in the type 2, they have, in the same method, they have loaded fluorescein molecules inside the mesoporous silicon nanoparticle, and additionally, they have modified that outer surface of the silica particle with triethylene glycol. So in type 1, there is no surface functionalization or modification, but in type 2, there is surface functionalization. So in type 3, they also loaded fluorescein molecule inside this mesopores. And instead of functionalization of the outer surface of that particle, they they capped these mesopores with gold nanoparticles. Gold nanoparticles that prevents from immature, like leaching of these chemicals inside these mesopore silica outside. So it prevents leaching molecules, and also silica is, as I said earlier, it's a low density material. So by capping these silica mesopores using a gold particle, it increases the density as well. So it takes like added advantage by doing this method. That is the type 3, a fluorescein loaded mesoporous silicon nanoparticle capped with gold nanoparticle. Then type 4, instead of fluorescein, they loaded beta estradiol. It's a chemical that should be needed to express, like, to see the expression of GFP, green fluorescent gene. That's what they say. So this chemical is needed to visualize this GFP expression. So they encapsulate. So they have used these different type of particles to different researchers. So they loaded beta estradiol inside, and same manner they clap the mesopause of the mesoporous silicon nanoparticle. So first, what they have done is they wanted to check whether what type of cell is preferentially uptake by the cells. So first, they have used type 1 and type 2 mesoporous silicon nanoparticles, which is loaded with the fluorescein molecule, which is the fluorescence molecule. So after incubation so for this process, they have used tobacco plants protoplast, that means the cells without cell wall cultures. So for that cultures, they using a gene gun, they delivered these type 1 and type 2 silicon nanoparticles to the cell culture, and after 24 hours, what they have observed is, so these small green-like particles are the fluorescence loaded mesoporous silicon nanoparticles. So I'm talking about these top images. So the red one of the red fluorescence that we can see from the first image is like autofluorescence vacuole in the plant cells. So they are autofluorescing. So instead of that, the green ones are the mesoporous silicon nanoparticles with fluorescein. What they have observed is none of these type 1 fluoresceins and loaded mesoporous silica particles did not take up by the cells. Instead, they have the cells preferentially uptake the type 2 mesoporous silica nanoparticles. So we can see inside of these red vacuoles, we can clearly see green fluorescein loaded mesoporous silica nanoparticles. So what they conclude is surface functionalization matters. So by doing the surface functionalization, they conclude that the type 2 mesoporous silicon nanoparticle can successfully apply to deliver proteinaceous or any to deliver chemicals successfully inside of the cells. So moving on to the next part, they have tried to optimize this with the DNA immunization. So for that, they have used they coated DNA with the type 2 mesoporous silica nanoparticles. So they have optimized the DNA coating ratio. So they have found out that the optimized ratio of DNA to mesoporous silica nanoparticles is 1 to 10 or lower than that. Yeah, under these conditions, DNA makes a stable complex with mesoporous silica nanoparticles. So with the using of that, they have successfully transfected cells. Like again, this is the tobacco plants chloroplast, and this is the left side image, second row. They tried to deliver these DNA directly into the plant leaves instead of plant cells. So what they have observed, they have successfully able to deliver these GFP gene containing DNA into the cells. So left side image, figure B, shows in the bright field, and C shows the UV filter or GFP under the GFP filter. So left side image, the D figure, it shows the immature maize embryo. So also they can successfully deliver these DNA materials into the maize embryonic cell as well. So using type 3 and type 4, that means fluoresceins inside encapsulated mesoporous silica nanoparticles, and beta estradiol encaps in loaded mesoporous silica nanoparticles. In this, the other part, what they have done is they have made so also after delivering into the type 3 and type 4 mesoporous silica nanoparticles into the cells, we need to they need to cap out. Gold particles should be released from the caps. That means the chemical should come out from the particle to make the necessary to see the gene expression. So beta estradiol has to come out from the mesoporous silica through the mesopores after the removing gold particles from the mesopores. For that, they have used DDT as the uncapping agent. So, but the summary of this right side image is the results that they have used type 3 in the presence of DTT and in the absence of DTT. So with the ash color bars shows with the in the presence of DTT uncapping agent, the fluorescent foci per the plant cell cotyledon that they used. So we in the absence of DTT, the black color bars shows the fluorescence foci, number of fluorescence foci. So what they have observed is for that they have made plants, tobacco plants with previously transgenic with the GFP gene. So what by delivering these type 4 mesoporous nanoparticle, what they are trying to do is to see the GFP expression. They said they need to have this beta estradiol chemical. So they with the DTT, they are going to uncap gold particles and release the beta estradiol inside the cells, and then it can show the fluorescence high intensity with these ash colors. But black ones, they couldn't observe high fluorescence intensity because without DTT, the beta estradiols cannot come out. So the expression or the fluorescence expression cannot see. So they proved that using DTT like uncapping agent, they can release the they can control the release of these molecules or chemical inside these particles as well. So they prepared mesoporous silica nanoparticles loaded with chemicals or preferred DNA with controllable release process, and can successfully transfer them into the plant cells or plant cells using a gene gun. So that's the conclusion of that research. For the next part, I'm going to talk about how the gene guns are used in human applications like transfect animal cells. So in this research, they use ballistic transfection of neuronal cultures using a handheld gene gun. So this is research they have done. What they have done is they had a commercially available gene gun. So they used that gene gun for one process. Instead, they have conduct another research by modifying that commercially available gene gun. So the parts that they have modified is the acceleration chamber inside this. They have changed in part A, it shows the commercially available Helios gene gun. Part B, it shows the modified version of the gene gun. So how they modified is they modified the acceleration chamber. So with a low pressure, they concluded that they can obtain like high pressure, high velocity of the particle. So this is from the figure A, the black color in the inset, it shows the spacer. Spacer is a part that is maintaining distance between the nozzle and the skin or the target area. So in the modification, what they have done is they remove this space, and also instead of this conical shape, nozzle, they replaced it with a new nozzle with a smaller diameter. By reducing this nozzle with a smaller diameter, what they have trying to do is they reduce the spread of the they reduce the spreading area of the particles on the skin by reducing the size of the nozzle. They limited, like they reduce the highly spreading area. Instead, they focus a small area and they try to penetrate these particles into a high depth. So that's how they modified, and this is the image that how they use gene guns to transfect these cell cultures. So in this image, this shows the black color one. The left image, the bottom figure shows that neuronal cell that was successfully transfected with the commercially available gene gun. So in that case, they have proved that they can successfully transfect and the gene transfect these microparticles coated with the yellow enhanced yellow fluorescence protein. So with the enhanced yellow fluorescence protein, they observed the successful transfection of the gene inside these neuronal. This green color one, left side, is the HEK, human embryonic kidney cells that they use to transfect this gene. And they observed that they were successfully transfer these genes, these microparticles inside to these cells. So the black color ones is a neuronal cell. For these two, they have used the commercially available gene gun. From the right side image, it shows the clavicle brain slice, tissues from a brain slice, and they try to deliver to proteins, like two proteins that can attach to subunits of the transmembrane in the neuronal cells. So in the by delivering two proteins together, they observed that they using that modified the gene gun, they were able to successfully deliver both of these proteins together. So inside the neuron cell, the cytoplasm is highly red in color. So the delivered particles with red fluorescence protein in like accumulated inside the cytoplasm, and the surface of these cells is like enhanced yellow fluorescence protein and also the white orange color mix. So that subunit A. So they confirm that the attach of this protein into subunits occur. That through that they observe that both of these proteins can co-delivered into the neuronal cells using a modified gene gun version. So, also researchers have done to bombard cancer cells, like deliver cancer anti-cancer drug directly to the cancer cells or cancer area. So they have tried to do that using a gene gun. So in this process, they have used porous silicon particles. Porous silicon particles in this figure, it shows how they used silicon wafer to make silicon microparticles through this process, like etching process. Then ultrasonication. Through all of these process, they finally produced porous silica microparticles, and through the surface adsorption, they encapsulate or loaded anti-cancer drug inside these mesoporous silica nanoparticles. So the figure A shows the gene gun schematic diagram of the process that they have used to transfer these anti-cancer drug into the cultured cancer cells. Also, it is their version of gene gun. They prepared a gene gun with addition of a laser tube. The laser tube that they used to focus the area, that targeted area. So with the help of laser, they focus the targeted area that we need to deliver the anti-cancer drug. And also using a vacuum tube, they also try to increase the pressure inside this gene gun. So helium tube, helium used as the carrier gas, and the gas used to accelerate the particles. So particle tube. In this particle tube, the mesoporous silica particles encapsulate a drug. Like the microcarriers are inside of this particle tube. So with the help of this gas, high pressure gas, the particles are coming through this tube with a high velocity and successfully transfect the cells that we wanted to treat. So figure B shows the SEM image of the mesoporous silica nanoparticles that they have prepared. And figure C shows the particle size variation, distribution of the particle size of the prepared porous silicon particles. And the figure D shows the depth achieved by using these particles. So using the figure D shows the depth achieved by using these silicon nanoparticles. So it's ranging from like 1,000 to 3,000 depth, micrometer depth they achieved using these silicon nanoparticles. So they compared that with the widely used gold nanoparticles using the same gun. All the conditions are same, but the particles are different. So this is the silicon particle achieved depth. The right side figure E shows the depth achieved by using the gold nanoparticle. So what they conclude is this optimized gene gun system is like they were able to highly increase the penetration depth of these microcarriers compared to the commercially available gene gun. So after loading the anti-cancer drug, the left side image, figure C, shows the first image cells only, that is the cells that tags with the fluorescence dye. Then they injected only the porous silicon nanoparticles to check whether there is any effect of these silicon nanoparticles for the cell's viability. For that, they didn't observe like much changes of the number of viable cells, live cells. And when they loaded anti-cancer drug into these particles, and after delivered those microparticles in the cells with the anti-cancer drug, they observed a less number of live cells. That means anti-cancer drug was successfully able to kill the cancer cells. So the bottom image with the red fluorescence one shows the dead cells. So with the initial control, there are like two dead cells can be observed in this image. So with the delivery of porous silicon nanoparticles, no cell death were observed, but with the anti-cancer drugs, large number of cells were dead. So they conclude that using this method, they were able to successfully deliver anti-cancer drug-loaded silica particles into the cancer cell culture. So they try to increase the dose, and also by increasing the dose, they conclude that by increasing the dose, they can increase the cell death as well. So that's the conclusion of that research. And also, as shown is this figure, they delivered these microparticles into agarose gel covered with the porcine skin. So porcine skin resembles like human skin. So they were, after the biolistic delivery of these microparticles use the modified gun, they were successfully able to show that there are fluorescence particle inside this gel. To go to the gel, so particles has to go through this porcine screen. So this figure B shows the skin surface of these porcine, and also figure C shows the fluorescence particle which can observed inside this agarose gel after the biodelivery method. So, also, some researchers have carried out to check whether the protein can be delivered using the same method. So that method called proteolistic. Proteolistics is a method that we are delivered protein intracellularly using this gene gun of mechanical method. So biolistic delivery of protein. So in same idea as the previous research, in this research they use gold nanoparticles, particle size is 0.6 micrometers, and they coated these particles with EGFP solution, green fluorescence protein solution, and they bombarded these microparticles into a onion epidermis cell. So in the figure B, it shows after the bombardment of these microparticles into the onion epidermis, they could be able to see the green fluorescence inside this epidermis cell. So they proved that using this biolistic method, proteins can also be delivered inside to the cells. Also, this blue color one figure F shows a blue color GFU, blue color protein, that cell fluorescence protein. So all of these images show they were able to successfully deliver proteins using the same methods. So in this research, in the same research, they used different colored proteins like red protein. Here, they instead of using this figure A shows the SEM images of the protein coated DNA gold nanoparticles, and with the red channel they observe this is after 36 hours. So after the coating of these particles after 16 hours, they observed measurable acceptable fluorescence remaining in these microcarriers. So after delivering them into the epidermis cell of the onion cells, they observed still the cells can be observed with the fluorescence. Fluorescence activity is still active, so protein can be successfully delivered into the cells. So figure D shows the pig ear they use as animal cells to prove that protein also can be delivered into the animal cells using gene guns. So in the figure E, the dark dots, they resemble the gold nanoparticles, and the blue color areas are the fluorescence molecules that they delivered using this biodelivery method. So the SEM right side figure shows the SEM images of the standard DNA. Left side image, right side image, DNA coded porous silica particles, and also the AR dried. So to coat these proteins onto these gold silicon nanoparticles, they use AR dry method or lyophilization method. So in the right side image, the bottom image, the third two images, like from the top, third one, these two shows the lyophilized protein. So the drawback is they observe the gold nanoparticles and the lyophilized protein separately. They are not well coated. So using that observation, they try to deliver these proteins lyophilized or air dried without the help of gold particles. So by drying this with air drying or lyophilized method, they dried these proteins, same proteins used for the previous experiments, and they will try to bombard these dried proteins indirectly without the help of gold nanoparticles. They try to bombard this with naked protein. So they were successful, and they were able to see the fluorescence inside the onion epidermis cell. So at some point, this red channel with the arrow, they observed some aggregation of fluorescence protein because of the some with this method, fluorescence molecule can aggregate on the surface and give a high fluorescence. So that's the reason for that. Also, they were able to see the green fluorescence as well. So, according to those researchers, limitation of these gene gun methods is relatively low throughput. That means we can achieve sometimes we cannot achieve like 90% of the delivery of the particles inside the tissue. So, relatively low throughput is a major drawback, and also cell and tissue damage is still there. It's kind of it's up to some point, still there is a cell and tissue damage. The cellular target cannot be controlled because we cannot control that it should be go to the cytoplasm, nucleus, mitochondria, or plastid. Also, the gene gun commercially available ones are highly expensive. So this highly expensive one we can overcome by making our own gene gun. And cell and tissue damage, people have tried to optimize this damage, reduce this damage. So researchers have been done to reduce this cell and tissue damage using by reducing the operation pressure and the particle size. But doing both of them, it also causes the minimization, the penetration depth. So if we reduce the pressure, it can cause the decrease of penetration depth, and it causes failure of DNA transfection. So therefore, researchers have tried to get the assistance of microneedle to increase the delivery of these gene into a higher depth. So the researchers have been done to, with the gene gun with the modified version, they attached microneedles at the end of this gene gun process. So with the pressure, first microneedles is impact with the skin or the cell and make a pores with the help of these microneedles, and through these pores, these microparticles with higher velocities can penetrate into a greater depth rather than using only the microparticles. So with the help of these microneedles, some researchers have to try to increase the penetration depth of these biodelivery method. So in this figure, the bottom one, it summarizes how it's gonna work. So A shows the simple diffusion process. If we have some chemical on the skin, it's not going to penetrate like in just to the few layers of the skin. But with the microneedle, they can go up to Langerhans cells. And also with the help of Langerhans microneedle and also a gene gun, they can go beyond the Langerhans cells. So they can like go more depth into the more penetration depth can be achieved using the microneedle assisted gene gun delivery. So just by the figure D shows the diffusion rate or the diffusion process by using only the microparticles by using simple gene gun biodelivery method. So with that, we can get some other assistance to increase this penetration depth of biolistic delivery method and enhance this process to make highly optimized vaccine delivery methods. So there is a future hope that we can use these gene guns successfully to deliver proteinaceous DNA materials or any kind of chemical successfully we can deliver into the humans, plants with a painless manner. So the advantage of this method, gene gun, is transformation of many different tissues and cell types, no limitations. So no need of binary vectors like bacteria or viruses. So high frequencies of code transformation can attain by doing, we can deliver two or more chemicals or genes together with these biodelivery methods. So simple transformation protocol is easy. So delivery of large DNA fragments to deliver mRNA, protein, growth hormone, insulin, any type of molecule that we want to express. So that is the end of my presentations. Thank you very much for listening, and I'm happy to answer any questions.